All right, good evening. My name is Holger Hintelmann, and I'm the Dean of Science at Trent University. So as we gather tonight, we respectfully acknowledge that we are on the treaty and traditional territory of the Mississauga Anishinaabeg. We offer our gratitude to the First Nations for their care for and teachings about our earth and our relations. May we honor those teachings. So on behalf of Trent University, it's a great pleasure for me tonight to welcome you to this year's Stairs Lecture in Chemistry. At Trent University, we are always be a proud member of the community, strive to be a strong partner, enhancing our community and culture of Peterborough. As part of this endeavor, we wanted to share our knowledge, such as we're doing tonight. It's just one of the ways we want to give back to our community. So tonight, it is all thanks to the generosity of Dr. Robert Stairs, Professor Emeritus of Chemistry at Trent University, and his wife, Sibyl. The Stairs Lecture in Chemistry is actually the first endowed lecture series in chemistry. And it's a biennial lecture which enables the university to invite distinguished scientists to speak on their research and to showcase the field of chemistry in general to our community. So on behalf of Trent University and the Peterborough community, thank you, Dr. Stairs, for your vision and dedication and for the legacy that you have left to Trent. Now I'd actually like to invite you, Dr. Stairs, to offer some words of welcome for our event this evening. Thank you.
Thank you, Holger, and thank you all for coming. I should, I suppose, properly begin by saying, dear friends, colleagues, ladies and gentlemen, I have a note. Yeah. Well, thank you for coming, whatever. And thanks also to Professor Emeritus for coming so far. And I hope he has a happy time here. From myself to Trent, for 29 years of friendship and stimulating employment, and from my late wife Sibyl for opening her eyes to new vistas, and Evharisto to Emily Vassiliadis for keeping us all on the rails. Sibyl and I discussed whether we should set up some scholarships or this lecture theory series, and we decided that Trent has a pretty good scholarship program already, and this is something a little different. And I'm very happy that it seems to be back in functioning again after the COVID hiatus. And I welcome you all, and thank you.
Well, we're almost at the main event. So thank you, Dr. Stairs. I'm Stephen Rafferty. If you don't know me, I'm the chair of the chemistry department. And it's my great honor today to welcome Professor Ian Manners of the University of Victoria to give the Stairs lecture for this year. This lecture tonight will focus on recent advances and applications of nanoparticles from the controlled crystallization of polymers. And seeing how many chemists are out here, most of you will understand what that means, or will soon understand that. I'll give a bit of Professor Manners' biography, and you notice there's an interesting oscillation here. He was born in London in the UK, and he earned his B.Sc and Ph.D. at the University of Bristol before conducting postdoctoral work in Germany and the US. And here's where the oscillation begins. In 1990, he joined the faculty of the University of Toronto as assistant professor, and 5 short years later, he made full professor. It's pretty amazing. And in 2001, he was made a Canada Research Chair, which is very prestigious. And in 2006, he returned to the University of Bristol to take up a chair in organic macromolecular and materials chemistry. In 2018, he oscillated back to Canada and was sort of Canada 150 Research Chair, which is even more prestigious than the Canada Research Chairs, and took up that position at the University of Victoria. And most recently, this year, in 2023, he won the Stacey Award from the Canadian Society for Chemistry for his distinguished contributions to chemistry in Canada. His research focuses on synthetic problems at a variety of size scales. We're talking about nanoparticles, we go from the very small moleculars, but also up to larger things. And spans a lot of things of interest to chemistry, catalytic main group chemistry and main group polymers, functional metallopolymers, crystallization-driven self-assembly of polymers, and more. He's published more than 800 papers, 4 books, presented more than 600 invited talks, and we're happy to have him for his most recent of these invited talks. So please join me in welcoming Professor Manners to give this year's STAIRS lecture.
Okay, can you hear me okay? You hear me okay at the back? Yeah. Okay, good. Yeah, I lost my voice about a week ago completely. So I was a bit of a panic since I knew I was coming to give these 2 talks. It's come back about 40 to 50%. So hopefully the microphone will do the rest. It's my very great pleasure to be here, privileged to be here at Trent. First time I've been to Trent University and yeah, having a wonderful visit, lots of very friendly people, and I can see it's a very exciting place to study, to do science and to do chemistry. And so it's really my pleasure to have the opportunity to talk about some of the work we do in our group in Victoria. And in this lecture, I'm going to talk about the largest project in our group. We have other projects, but in this lecture, I'm going to talk about making nanoparticles. It was actually going to be from polymers. And we're going to use crystallization to control how we form the nanoparticles. And I should say this was kind of discovered by accident about 15 years ago. And we've developed it ever since. And what's really exciting is that initially it was very fundamental studies trying to understand how crystallization affects nanoparticles. But now what's really exciting for us is that we're starting to move to applications. In this lecture, I'll give you a flavour at the end of some of the recent work we've done where we're looking at applications of some of the nanoparticles we can make. So we call this crystallization-driven self-assembly. I'll explain what this is in a few slides' time. And very importantly, we're able to develop this into, again, I'll explain it fully, a seeded growth method, which allows you to control particle sizes very precisely. And this method we call living CDSA. Living because you can add more polymer and grow the particles in a kind of living way. It's very controlled. And this method is kind of unique for making nanoparticles with controlled dimensions, as I'll explain. I'll talk about the scope of this method, how you can use it to make different types of particle. And then at the end, I'll talk about some applications that we've been looking at. And I'll probably have to stop a few times for some water. So please bear with me. So you can classify different structures on different length scales. I mean, up here are length scales we're familiar with everyday life. And if you go down to atoms and molecules, you're down below 1 nanometer in size. Polymers are long-chain macromolecules. So something like polyethylene or polystyrene are long chains of carbon atoms. And these are bigger than normal molecules. These are up between 10 nanometers and 1 nanometer in size. But there's a size regime here between about 10 nanometers and 100 microns, where really biology reigns supreme in terms of making structures. So the structures we usually think about in this size regime are biological materials. Now, chemists are very good at making molecules. They spend a lot of time over the last 100 to 200 years making molecules. There's still things to learn, but chemists are generally very good at making molecules. They're also very good at making polymers, but chemists generally are not good at making structures in this size regime. So somehow you've got to make larger structures here. These are orders of magnitude larger than the normal structures you make in chemistry. So how do you make these types of materials? That's going to be the subject of this lecture. And we're interested then in new methods, synthetic methods for making structures in this size regime. So these are really nanoparticle size regime here. And if you're going to make particles, you want to be able to control their size. You want to make samples that are uniform. And the technical term for this is monodisperse. This means if you make a sample of particles, they're all the same size. You don't have some large ones and small ones. Large particles and small particles can behave differently for an application. So this is not good. So you want to have very uniform samples. You also want to make structures where you can control functionality. So you can put different types of functional groups which allow you to control properties on different parts of a particle. So this is also important. And we'll see each of these addressed later in the lecture. So the magic method for making structures in this size regime from smaller structures is self-assembly. So you make smaller structures and get them spontaneously to self-assemble into larger structures. And here's a very good example of this. It's really well known, and it's an example we all use when we're washing up, or at least your dishwasher uses it. And that is using detergents or soaps. And these are molecules which are called amphiphilic. This means they have a polar part, which is hydrophilic. So this bit, the purple bit, likes, for example, likes water. And the other bit is hydrophobic. So the other bit hates water. And it likes oil and grease and things like this. So if you take these molecules, which you can make very easily, you can buy them. And when you buy a detergent or soap, this is what you have. Here's the polar bit. Here's the hydrophobic tail. If you put these in water, this bit likes the water. This bit hates the water. So what happens is this hides in the middle and this goes around the outside. And you get what's called a surfactant micelle. Here's a cross section. So these are core shell nanoparticles. And this is what you have when you have a soapy solution. The size of these structures is such that they can scatter light. This is why your solution looks soapy. So you have a core here, which is hydrophobic, and then around the edge, you have a shell or corona, which is hydrophilic. And the beauty of these is if you have some grease or dirt, if you try and wash something with just pure water that's greasy or dirty, of course this doesn't work. But if you have these structures around, these have a hydrophobic pocket that will dissolve grease and dirt. And so when you have your greasy object and a solution of these micelles, as you do in a soapy solution or a detergent solution in water, the grease and dirt will go into the interior here, and you clean whatever object you're trying to clean, clothes or plates or whatever. So these are incredibly useful structures, but they're only stable in solution. These structures fall apart if you remove the water. They basically just fall apart. And the reason for this is the hydrophobic tail is rather short, as you can see here. And this doesn't give this enough stability to exist in the absence of a solvent. So what you can do is make the hydrophobic tail much longer and use a polymer. So instead of having a hydrophobic tail that's maybe 10 or 15 atoms long, you make one that's hundreds and hundreds or even thousands of atoms long. And then you play the same game. So what you have is, this is called a block copolymer now. So instead of a surfactant, you've now got a hydrophobic part in blue, a hydrophilic part in orange over here. And these are long chains. So when you put them in a solvent that is good for the orange part but bad for the blue bit, you get micelles. But these now are much more stable. And in fact, if you remove the solvent, you can actually visualize the micelles by electron microscopy and other microscopy techniques. And throughout the lecture, I'll show micrographs of micelles, and these will be obtained by mainly transmission electron microscopy or atomic force microscopy. You can get micelles of different shapes, and people have been very interested in these structures for lots of different applications. Since they're much more stable than surfactant micelles, you can think about using them to do lots of useful things, apart from cleaning the dishes and cleaning grease off of clothes. So people have been looking at them in particularly for delivery vehicles for drugs in nanomedicine. One way to deliver a drug is to have a shot in the arm. Of course, when you do that, initially you get a very high concentration of the drug. And if a drug is toxic, this is bad news, then it goes throughout the body. So you might only want the drug in a small part of your body to treat one organ, but you're going to get it everywhere in your body. And then rapidly, the concentration of drug will go down as it's excreted from the body. But another way to do this is to have a delivery vehicle. That is to deliver a drug through a therapeutic agent. So you can actually put drug molecules into the core of these structures, or in this case into the hollow core, and you can put targeting groups on these structures so they will target different organs, and then you can control the diffusion of the drug out of the nanoparticle to the organ to treat a particular disease. So these have a lot of interest as drugs in nanomedicine. One of the problems is one of the most useful shapes is a cylinder. These shapes, they exist much longer in the body. The other shapes tend to get excreted from the body very quickly. But cylinders tend to stick around much longer. So in principle, they're much better for uses for drug delivery applications. But unfortunately, cylinders are very difficult to make, usually using this method. Usually you get spherical structures or vesicles. These are difficult to access. So many of the applications are shape dependent. They depend on what you can make. And here's some of the applications that have been looked at nanoreactors, using them as reinforced composites. So just like you use fiberglass to reinforce a composite, you can take nanoparticles, again cylinders are particularly good, and use them as nanoscopic reinforcing agents. And they're very, very effective for improving mechanical properties. You can also think of using them as nanowires, and I'll show some examples of this later. So as I've mentioned, these are generally much more stable than micelles formed by molecular surfactants. So as I mentioned, spheres are the most common here. Others are difficult to form. 1D structures like cylinders. These are very promising, as I mentioned. They have good circulation times and cell uptake rates. But these are difficult to make, so this is a problem. You really want to be able to make these structures much more easily. Another problem is usually there's no control over the size of these structures. So if you make cylinders, you can't control whether they're short or long, and your samples have a mixture of lengths. This again is bad for applications because it basically gives you a mixture of properties. So that's another problem that we want to address. What you'd like to have is uniform samples, or what I explained as monodisperse samples. So what I'm going to talk about is work on these types of systems. These are called block copolymers. I'll use this abbreviation, BCP. So they're like molecular surfactants, except they're made from polymers. So you have a hydrophobic part of the block copolymer in blue here, and then a hydrophilic part. And you can self-assemble these polymers in water or also in other solvents, as long as the solvent only dissolves one of the blocks. And we'll see lots of examples of this. So this is what we're going to discuss. And specifically what I'm going to talk about is how the block that forms the core actually affects the self-assembly. And what we found is that if you have a core forming block that can crystallize, this dramatically affects the self-assembly. And it turns out virtually all the work that had been done until we got involved in this field, along with our collaborators, nearly all of it had been done with core forming blocks which will not crystallize. So we kind of stumbled into, really by accident, into a new area. And it turns out crystallization really affects self-assembly. So let me illustrate that. And this is where we started. The polymer we used as our core forming block were polymers with this type of structure. And if you're a chemist, you'll recognize this as ferrocene. This is a silicon with two substituents. And this particular polymer has two substituents on silicon that are identical. And this was actually the polymer that we first looked at as the core forming block in our block copolymers. I'll call this polymer, this has two methyl groups. These polymers are called polyferrocenyl silanes. And I'm going to refer to this as dimethyl PFS, or just PFS, in the following slides. So let me tell you about this polymer. It's an orange polymer. It forms nice transparent yellow films. But very importantly, it's a crystalline polymer. What I mean by that is these polymer chains are so symmetrical, they can pack into an ordered crystal lattice. And you can actually grow crystals of this polymer by heating it in, dissolving it in hot solvents and then cooling it. And these are crystals of this polymer. And has a melting point of 145 centigrade. Now, if you change the substituents of silicon so they're different, you see a dramatic change in the properties. And I'll show you that in a moment. You might think about how these polymer chains pack into a crystal. This is the packing that you see. This is a model pentamer, so this is 5 units of this repeat unit here, which you can crystallize, and we have a structure of this. But this is a model for the polymer. If you can imagine these chains continuing, this is how the polymer chains pack into a regular crystal lattice. Now, if you replace one of these methyl groups by a different substituent, by an ethyl group, so these are now different, these polymer chains are now unsymmetrical, so they can't pack into a crystal lattice. And you get a total change in properties. This is what this polymer looks like. So this is called an amorphous polymer. It's unable to crystallize, in contrast to this one, which crystallizes, and you can form crystals, and you get very different properties. You can form nice plastic films of this material. So this has a melting point, this one doesn't. So what happens when you do self-assembly with this as a core-forming block? So these are the first experiments we did back in, this would be about 1995, we did these experiments for the first time. So here's our block copolymer. This is our dimethyl PFS block that can crystallize. We're going to use this as a core forming block. And this other block is a polysoloxane block. And this block will dissolve in solvents such as hexanes. Whereas this block, the PFS block, is insoluble. So when you take this block copolymer, dissolve it in hexanes, you expect to form micelles where this forms the core and this forms the corona. And what we were expecting was to form spherical micelles. This is the most common morphology or shape you see for micelles when you self-assemble block copolymers. But to our surprise, what we got were these cylindrical structures, or what we might call fiber-like micelles. And so this is the structure. This PFS block that crystallizes forms the core, and the coronal chains here are on the outside, form the shell. So why do you get these fiber-like structures? Well, if you assemble this block copolymer, instead of at room temperature, you do it at 150 degrees. That's above the melting point for this block. And so at 150 degrees, if you do the self-assembly, this block doesn't crystallize because it's above the melting point. And remarkably, what you then get is the spherical micelles that we expected. So this tells you that crystallization is affecting the self-assembly. If you let this block crystallize, you get fiber-like micelles. If you don't, you get the spherical micelles you expect. And you can demonstrate this further. If you make the same block copolymer, but we now have the PFS block with different substituents, so it can't crystallize, even at room temperature, this forms the spherical micelles. But this block can't crystallize at any temperature. So this is the shape micelle you get as illustrated here. So what this shows is, and really for the first time, was the crystallization of the core forming block can affect the self-assembly. So we call this crystallization-driven self-assembly. Crystallization affects the shape you get. And what's nice here is spheres are very easy to make, but fiber-like structures are very difficult. And you get a pure fiber-like morphology here. They're all fibers. Now the problem here is you get fibers with different lengths. You can see short ones here, long ones here, ones in between here. So a key question is, how can you control the length? And to do this, we developed a seeded growth method for doing crystallization-driven self-assembly, which we call living crystallization-driven self-assembly. I'll show you how this works. the first step is exactly what I showed you on the previous slide. You take a block copolymer such as this one, where you have a crystallizable core forming block. This block forms the corona or shell, and you self-assemble this in, for example, hexanes. And this gives you fiber-like micelles, as I've shown you, where they're all different lengths. There's no control over length here. Now, I mentioned the core is crystalline. You can show this by lots of physical techniques. I'm not going to go into any detail on this, but you can do X-ray diffraction, electron diffraction. You can study the core of these micelles, and you can show it's crystalline. So these fibers you see here are polydisperse. They're not uniform. So what we want to do is to make uniform fibers so that you can tailor them for different applications. And we want to control the length so that you can make different lengths for different applications. So how do you do this? Well, the key is to take these structures here and sonicate them using an ultrasonicator, and you break them down into little seeds. So these are about 10 to 20 nanometers long, you can see here. I think it's easier to put it here if you don't mind. Cheers. So once you've got these seeds, you can then take the seeds and add more polymer to the seeds. What's remarkable when you do this is the polymer grows from the seeds and you get controlled growth. So each of these seeds acts as an initiator for the growth of fibers. Each seed gives you one fiber. So if you control the ratio of the polymer you add to the seeds, you can control the length of the fibers. And this is illustrated here. This is the ratio of the seed, of the polymer we add to the seeds. So here is 5, here is 10, here is 20, here is 40. You can see the fibers grow in a very controlled way, and you're making uniform or monodisperse samples. This is the length distribution, is very narrow distribution, and you control the length here. And the reason this works is these seeds are crystalline, and when you add the polymer, the PFS block, which forms the core, crystallizes off of the seed, and you get a process called epitaxial growth, where you propagate the crystal from the seed and you grow the fiber. So it's like growing a crystal. That's really what you're doing here. If you plot the length against the ratio of the polymer you add to the seed, you get a straight line. So it's a very controlled process. By choosing this ratio, you can choose the length of the fiber that you make. And as I mentioned, you get very narrow distribution. And as I said, you get this epitaxial growth of the unimer from the terminus of the seed, and you get growth to give you the fiber. So this is a living growth from seeds by the addition of further block copolymer. So we call this living crystallization-driven self-assembly. And it's a controlled seeded growth method for making these types of structures. So this is how you do it in the lab. You take a solvent, which is good for the corona forming block, poor for the core forming block, and you add the polymer to the solvent, and it will spontaneously give you fiber-like micelles with different lengths. You then sonicate these. So you put in a sonicator into the solution. This breaks them down into seeds. And then you take the seeds and you add more polymer to the seeds, and then you get the controlled growth or living CDSA process. to give you your sample. So you can do this in air or with wet solvents. It's a very easy process. And you can also scale it up, which I'm not going to talk about, but that can be done. And you can make all sorts of structures using this method. Let's imagine you had a polymer that has the same core forming block, but a different corona forming block. If you now add that to these structures, you grow off the end of your new polymer and you generate a block structure where you have different corona at different places. Here's your original seed from this step. This is what you make in the third step. And now you've grown and more polymer off the end. And you've got different corona here. So this is called a block comicelle. This means you can put different chemistry on different places on the structure. And this is very important for different applications. And we'll see some examples of this later on. Now, here's one of these polymers. Here's the crystallizable core forming block. This is the shell or corona forming block. And you can make these with groups. These are called vinyl groups. And what's beautiful about those is you can functionalize them very, very easily using very convenient methods. So you can take these molecules, these are called thiols, and you can buy an enormous variety of these structures. And if you add light, so you shine light on the solution in the presence of this photoinitiator, you get a photoreaction where this thiol adds to the vinyl group and you tag it on there. And this is incredibly useful because it means that you can functionalize the corona forming block with anything you like really here. And this enables you to make fiber-like micelles where expressed on the surface of the corona is any functionality you want. And we use this for an enormous number of applications. So you can add on all sorts of different types of groups here. If you're looking at drug delivery applications, you can add targeting groups, which mean these structures will head for a certain organ in the body. You can add different color fluorescent dyes on here, and so you can make polymers where the corona gives you different types of fluorescence. And here's an example of this. You can do this seeded growth method where you add different polymers where the corona has different color fluorescent dyes attached. So you can add a polymer with a red dye, that's ads. Then you can add a polymer with a green dye, and then finally one with a blue dye. So you can make this block structure with multiple fluorescent dyes. And this is a fluorescence micrograph in solution of what you can make. And when we saw structures like this, we were very excited because no one could do this. In the past, people couldn't even control the shape of the micelles very well that they can make. But now we can control shape, we can make structures any length we want, make the sample such that they're all the structures are the same length. And we can even make block structures where we can functionalize as we wish in different places. So as a technology, this was very exciting to us. So this was about 2014, and this is when we started to think about applications. You can do lots of things with living crystallization-driven self-assembly. I'm not going to go into tremendous detail in this lecture on everything you can do, but I'm going to show you some of the structures that you can make. You can make seeds that grow only in one direction. The seeds I showed you so far grow in two directions. We've been able to make them so they only grow in one direction. And then you can make block structures. Instead of being structures that are centrosymmetric, so like B, you can now make AB structures. And here's examples of AB structures. Here's some more. These are ones with fluorescent dyes. The ones I showed on the previous slide these are centrosymmetric. So you have a plane of symmetry here because the seed grows in two directions. Here we use seeds that grow in one direction, so they're no longer centrosymmetric, they're non-centrosymmetric. You can make branch structures, as we've shown here. You could also make these structures so they themselves will self-assemble into micelles. So you can make these structures where the middle here is hydrophobic and the ends are hydrophilic. So this is just like one of the amphiphilic surfactants that I showed you earlier, or amphiphilic block copolymer, in that because there's a polar end and a non-polar end, these will self-assemble in solvents that are selective for one of the blocks. If you put this in a solvent that is a poor solvent for the hydrophobic center, but a good solvent for the hydrophilic ends, then you can get these to self-assemble into supermicelles. So this is a micelle made from these micelles. So it's a micelle made from micelles. And now you're getting up to really long length scales. These are multiple micrometers in length here. You can play this game in many different ways. You can make structures where you have a middle that is able to undergo hydrogen bonding. Hydrogen bonding is an important force for building up structures. It's used in biology. It's also very important in water itself for bonding between the molecules. And you can have hydrogen bond donors and hydrogen bond acceptors. And you can build up block comicelles where you have hydrogen bonding acceptors in one part and hydrogen bonding donors in the other part. So these, when you stick them together, they will actually aggregate, as you can see here. So you can build them up into really elaborate structures by having hydrogen bond acceptor and hydrogen bonding donor regions in these structures. If you put a hydrogen bonding acceptor region in the middle of one of the fibers and the hydrogen bond donor in the middle of the other, when you mix them, the hydrogen bond acceptor and hydrogen bond donors come together and you get cross structures, as you can see here. So you can easily make these cross structures by different methods. Here's a sample of these. So you can see the yield is basically 100% of these cross structures. Here's a micrograph of these cross structures. And the two fiber-like micelles lay over the top of one another. And so you can see, this is called a height image by atomic force microscopy. Here's the height trace. You can see where they cross over is much higher because they're crossed over kind of like this. Now you can use these cross structures to perform further living CDSA experiments. You can actually grow off of these structures. I'm not going to go through this in a lot of detail, but you can do 4 more living CDSA steps. And this allows you to make these structures. These are windmill micelles that you can make. So this is in seven sequential steps. And these structures you can see by electron microscopy. But these are so big, it's difficult to visualize more than one by electron microscopy. So a good way to visualize more of them is to use fluorescence microscopy. And so what you do is you build in fluorescent dyes into this structure. And then you can use fluorescence microscopy to visualize them in solution. And so we put green fluorescent dyes in the end and red fluorescent dyes along here. And so you can visualize them now in solution. And these are very stable structures. We've made samples where all the dye is green and samples where all the ties red. And then when you mix them, we wondered whether there would be any exchange, but actually very, very stable. So the green windmill micelles remain as green windmill micelles, and the red ones remain as red ones. So far I've talked about making 1D structures by living CDSA. You can also make 2D structures. And these are very, very difficult generally to make normally by self-assembly. So we're very excited that we could make 2D structures as well as 1D structures. And the way you do this is you make, again, a amphiphilic structure, so one that has a charged part or polar part and another part that's non-polar. And we use a polymer that will crystallize for the core forming segment, and then we have a charged group which dissolves in the solvent, and this is what allows you to make colloidally stable micelles, so they're stable in solution. And so again, when we started this work, we used this polymer with the two methyl groups on silicon, which will crystallize, and this is our charged group, a phosphonium group at the end. And then what we do is we make 1D seeds by living CDSA, and then we add this polymer to them. And when this adds, it crystallizes off of the ends and then surrounds the seed, and you get a structure, 2D structure like this. And you can make 2D structures where you can control the dimensions, and they're all the same size. So here's some examples down here. So these are electron micrographs. This is TEM, transmission electron microscopy. And you can see the original seed in the middle, and then this grows around the outside. This is AFM, atomic force microscopy, which allows you to look at the height. And this is the height as you go across here, this blue line. And you can see as you grow across the seed, it's much higher than the rest of the structure. So again, you can control the dimensions of these 2D structures by the ratio of the polymer you add to the seed. Here's the ratio, 4, 12, 20, 32. You can see the structures getting bigger. If you plot now the area against the ratio of the polymer you add to the seed, you get a straight line. So you can control the area. If you add polymers, that you can do this with polymers with different corona-forming blocks that have different chemistries. For example, they can have a different fluorescent dye attached. You can again build up block structures, but now they're two-dimensional block structures. So this is a 2D block co-micelle where we've done sequential additions of different polymers to the seeds. And here's an image by atomic force microscopy. If you use different fluorescent dyes attached to the polymer, then you can show the different blocks you're forming now in 2D, and you can see the different regions which arise from the fluorescence. You can also use a corona-forming block that, for example, will bind to nanoparticles. So you can use a corona-forming block that will bind to silicon nanoparticles, and then when you add silicon nanoparticles, they selectively add to those regions. And so you can spatially, selectively add nanoparticles to different regions of your structures. So you can build up block structures. This is 7 blocks, heptablock. And the other thing we've been able to do is actually dissolve out the middle of these structures. And this allows you to make hollow structures. This is a middle that's been removed from the center of one of these. But you can make these hollow structures as well. So you can make a huge range of different structures with great control. And so this is from 2016. And what we've really done since then is try to generalize this to different polymers and also to develop applications. And so what I'm going to show you in the last part of the talk is just some of the applications we've been looking at. In tomorrow's lecture, I'll talk some more about applications and I'll talk about some different applications. But this is the model we have for our living CDSA. But at this point, all the work we've done is with one polymer, this dimethyl PFS polymer. Now, many polymers are known to crystallize, so you'd expect this method to be generalizable to many different polymers. And we and other groups around the world have used this now for many different polymers. And these are some of the polymers that have been used. These are some of the different groups around the world that have done this. This is a polymer made from an organic polymer called polythiophene. It's particularly interesting because it's an electrically conducting polymer. And this allows you to make 1D structures that actually conduct electricity in a very controlled way. So this is a block co-micelle we've made with a core forming block from P3HT. And I'll come back to these a little bit later and I'll show some applications of these. You can also use other types of structures, not just high molecular weight polymers. You can also use large molecules that will form stack structures, like porphyrins, benzochroranines, perolindiamides, many others. These are molecules that stack into one dimensions. And if you add a polar group to the edge, so they're amphiphilic, you can also do living CDSA. And we and other groups around the world have done this as well. So this is another extension of this living CDSA CD growth method. Okay, so in the last few minutes, I'll just quickly go through some of the applications we've been looking at. And this is all really recent work. We've only now really started to look into applications, but there's a lot of other groups working on this as well. So it's an exciting area to be in. As I mentioned, one application for my cells is drug delivery. Now, 1D structures are particularly good for drug delivery because they have a longer retention time in the body. And as I'll show you, they're also much better at, for example, penetrating tumors, which will be good for therapy for cancer, for example. And we've shown this by making a lot of different 1D fiber-like micelles with different lengths. So we made them with different lengths. We also made spherical structures, and we tagged on them fluorescent dyes so that we could visualize them penetrating tumors. And these are the images of the tumors that we were looking at. And the key point is the more fluorescence you see means that you're getting more penetration of the tumor. And what you see is the fiber-like structures are much better than spherical structures for penetrating the tumor. But secondly, if you have fiber-like structures that are 80 nanometers long, these are much better than the other lengths. So this allows you to make structures, 1D structures that are better at penetrating tumors, and also to tune the length so that they are better at penetrating tumors. So these should be much better for delivering drugs, for example, for cancer therapy, to these particular breast cancer cells. So we think this is a significant result. This is a collaboration with groups at the University of Toronto. But this tells us that these structures should be particularly promising, for example, for cancer therapy. We've also started looking at the actual delivery of drugs to cells. And one of the most exciting types of drug delivery is actually to deliver genes to cells. Gene therapy, I'm sure you've heard of, a particularly exciting area. It's really revolutionizing nanomedicine. But somehow you've got to get your gene into a cell. How do you do that? If you just add DNA, if you inject DNA, generally it won't go into the cell. It won't go across the cell membrane. So you need to help it across the cell membrane. And so it turns out that these 1D fiber-like micelles are really good for penetrating cell membranes and actually delivering drugs, and in this case, genes, to the cell. So again, we made 1D structures with lots of different lengths. We also made spherical structures. And we use this polymer here. This is a biocompatible, crystallizable polycarbonate, which we use for the core forming block. And the corona is a polyamino methacrylate. The reason we use this is that if you put it in water, it will protonate, and this means it becomes positively charged. DNA, of course, is negatively charged, so DNA sticks to these fiber-like structures, and you can put a lot of DNA into the shell of these structures. And these are actually some of these structures with DNA bound. So we made lots of different lengths, and we use a plasmid DNA that, when it's delivered into a cell, it expresses luciferase, which gives you luciferin, which is a green fluorescent molecule. It's actually the one that's used for the fluorescence from fireflies. And then to judge the success of the incorporation of the DNA into the cell, the transfection efficiency, you can use fluorescence then to measure the transfection efficiency. So we did this using brain cancer cells. We made fiber-like micelles with DNA bound. These are called micelle plexes. We made them with different lengths, and we also made spherical structures to see how they behave. And what you find is, here's the shapes down here. Short fiber-like micelles are really efficient for gene transfection. So if you want to deliver DNA to cells, this is a great way to do it. Currently, one of the commercially available ways of doing this is called lipofectamine, and this is much more efficient than lipofectamine, so it completely outperforms the current really gold standard for gene transfection. So we're very excited about these results. This is very, very recent. End of last year we published this work. So nanomedicine is an area we're working in. Another area that you can work in, if you choose a core forming block that will conduct electricity, is to make, for example, nanowires. So we use this polymer made from a core forming block, which is a crystallizable polythiophene, and then we have a corona forming block, which is a polythiophene that will not crystallize. And you can assemble these into fiber-like micelles by living CDSA. And then you can use methods to actually look at individual fibers, and you can measure the current along a micelle. And this is the tunneling current along these micelles. So these are fiber-like micelles that we place on a surface, and then we use this method where we use a tip that can measure the current between the tip and the substrate, which is gold, and you can see the tunneling current. So these really conduct electricity. And then we wanted to see if we can make devices out of them. So you can flow align these in concentrated solution. If you squirt them out in a direction, they will move with the flow of the fluid and then dry so that you get orientation like this. And we're able to make field effect transistors out of these structures. And they actually behave like normal field effect transistors. So you can make structures of interest in optoelectronics as well using living CDSA. And I'll talk about this some more tomorrow in tomorrow's lecture. Finally, one application that we've been looking at is to functionalize services using living CDSA. And what you can do is you can spin coat seeds that you make by sonication onto a surface, as you see here. Now they're stuck to the surface. If you now add polymer to the surface, it will grow from the seeds, and you can grow brush layers from the surface. And you can visualize these brushes by the method of atomic force microscopy, which you can also do in solution and in the dry state. If you do it in the dry state, these brushes all collapse on the surface, and that's what you have here. But if you visualize in solution, they're all pointing upwards, and that's what you can see here. So these brushes, you can do lots of interesting things with these structures. One thing you can do is control surface properties. So if you have a corona-forming block that is hydrophilic, this gives you a hydrophilic surface. So if you measure the contact angle, it's very low, it's about 67 degrees. But if you then grow another polymer from this that has a hydrophobic corona, this gives you now a surface that is hydrophobic and the contact angle is much higher. So you can control surface properties. These brushes can be used for lots of different applications. I'll just show you one application that we've been looking at, which is where you can attach nanoparticle catalysts to the surface by binding them to these brushes. So you can take a surface or a capillary and you can add the seeds to the capillary so they attach to the surface. You can then add polymer so it grows from the surface. So you have a brush layer on the interior of the capillary. You can then add nanoparticles which stick to the brush layer. You can do this by two methods. You can make the nanoparticles first and then just add them and they will stick. Or you can do a two-step in situ reaction where you start with a metallic salt and then you reduce this down to the metal which forms the nanoparticles and then they stick to the brush layer. So here's an image of these brush layers with particles attached. The reason we do this in capillaries is because there's a lot of interest in doing catalysis with combining catalysis with flow chemistry. So you can do catalysis under flow by taking these capillaries and then you can put different catalysts on different capillaries and then do sequential reactions by just sticking the capillaries together. And that's what you have here. You have different capillaries stuck together. You just flow the reactants through the capillaries and you can do catalysis. I don't have time to go into this in great detail, but you can do all sorts of organic reactions, coupling reactions, photolytic reactions. You can do multi-step cascade reactions where you put a different catalyst in each capillary and then you flow through your reactant you do a different catalysis at each stage. So you do a multi-step cascade process. And this turns out to be a very interesting way of doing catalysis. We published some results on this in 2021. So again, it's very recent, but it's another area that we've been working in. So let me summarize. Hopefully I've explained the idea of using crystallization to control self-assembly. This was discovered completely by accident, but it allows you to really control self-assembly in a very different way, in a very controlled way. And it allows you to develop the seeded growth method of living crystallization-driven self-assembly, which allows you to make structures on the 10 nanometer to 10 micron length scale in a very controlled way. As I mentioned at the start of the lecture, this is a length scale that normally biology is very good at, but chemists aren't very good at. So we're very excited that this is a good method for making structures in this size regime. You can make 1D structures, 2D structures, and also more complex structures. You can do this with a wide variety of different materials by controlling the nature of the core forming block and also the corona. So you have complete control over the properties of the structure that you make. And at the end, I talked about some applications. I didn't have time to talk about all of these, but I did talk about promising results in nanomedicine, for drug delivery, catalysis, nanowires, a little bit about optoelectronics and surface functionalization with micelle brushes. Let me thank the people that did the work. These are the people that contributed to most of the work that I described in the lecture. I won't go through them all in detail. Many are now professors at different places around the world, some in Canada. We've got great collaborators. I should mention in particular Mitch Winnick and Gerald Gearing at Toronto, who have been collaborators from the very beginning on this project. We've got many other collaborators around the world. These are the people that fund us. Here's my group in Victoria. There's a few people missing. There's these individuals. So this was 2021. I also have a position in China at Shanghai Jiao Tong University, where I co-supervise 6 or 7 students. This is from a Zoom meeting in 2020. So I'd like to thank them. Huibin Chu is the professor in Shanghai. And finally, I'd like to thank you for your very kind attention. Thank you very much.